is another suspect who we don't need to talk about. And here is the oral smear slide. Non-sperm fraction, that's the profile of the victim, or is thought to be. And this is what incriminated Mr. Puckett, the oral smear slide sperm fraction. Okay, so let's look quickly at it. You know, he's got a 14 and a 16 allele. You heard about it this morning. And there is a 14 and a 16 in there. There's also a 15, which tells us it's a mixture. Right? There's somebody else's DNA is in there. The lab's theory is that that other person was the victim, because the victim has a 15, so it could be her 15. Although there's an obvious problem with that theory in that none of the victim's alleles are found at any other locus. So just that locus, right? Which is, you know, it could happen, but that's a bit problematic, right? I mean, the other theory is that this is DNA of, the victim's DNA isn't here, and then the 15 must be from a second person. Like maybe she had sex with two different people and one had a 15. All right. So now, and interestingly enough, that changes the statistical computations. To get the frequency of 1 in 1.1 million, the lab has to assume that the 15 came from the victim, right? And so that they computed the 1 in 1 million, assuming that the perpetrator either was a 1416 or a 1516 or a 1616. Right? So there's a restricted number of possibilities to get into the 1 in 1.1 million. If you assume that there might be two men rather than a woman and a man in the DNA, which seems to me it's sort of possible, right? Then the number changes from 1 to 1 in 170,000. Right, that's just because that means that the true perpetrator could be a 16, 16, 15, 15, etc. I mean, there are further issues. Like here, for example, they looked at this and they assumed there's a 14. The true perpetrator has to be a homozygote. And he is, okay, so he matches. Now here they found just a 28. And so, do they assume the true perpetrator must be a homozygote? No, because he doesn't match, right? So here there's an asterisk, which means they assume that one of the two alleles has disappeared. Well, why don't we? What would happen to the statistics if we assumed that the true perpetrator might have had a second allele here? That changes the statistics further, and now we're 1 in 9,400. All right, so. All right, so things change dramatically depending on how you interpret it, and the basic interpretation of the strength of the evidence is contestable, let's say. All right, and yeah.
So do they usually do this where they do the analysis while looking at the reference sample, so that they're because the interpretation that they assume the one male contributor, it almost seems like they made that assumption because they were looking at the crime scene sample.
Well, yeah. No, in this case, they made some initial assumptions before they searched the profile through the database, and we know what assumptions they made. We know what they searched through the database. So at least on the time when they got the hit, I don't know if they did some earlier, searches, but when they got the hit, they assumed that the 15 was from the female victim, and that everything. They made that assumption. They assumed a missing allele here, no missing allele here. That was what they searched through the database. And you can view that as kind of a blind read, and I'm usually okay with blind reads. Just in this case, I don't know if they did an earlier blind read, but we have a DNA analyst. Do you have a thought?
As a scientist, I think it's also possible that because it's a degraded sample, and I'm just speculating on their behalf.
Yeah.
That because the D18 locus didn't work, that could have been a smaller peak that
right.
Didn't meet the criteria for calling it a real peak.
Yeah, there are.
And a second peak at the 21 that was also a possible peak.
Yeah, there were. I actually. Yeah. No, I think that's true. And where they have the asterisk.
Obviously a mixture.
Yeah.
Because it has 3 alleles at D3, so
Yeah.
have to have 2 different.
It's true. No, I looked at the autorats, and where they have asterisks, it does indicate lower level peaks below their threshold. If the peaks are below a certain size, that is the alleles, they're not deemed reliable, so they don't report them. So the asterisks indicate that there were something there. Now, but if you look closely, some of them match Mr. Puckett. Yeah, they may or may not be real, and among those, some of them actually match Puckett and some don't, right? So one can look at these below threshold peaks as providing providing additional support for the second contributor theory also. If you want to treat unreliable data as reliable, then it then has to be interpreted fully as well. So this is just an example, but a recent one of somebody who was convicted of a real crime. Who I think there's a good argument that can be made that this guy might not be guilty, although the jury certainly thought he was based on how it was presented. Now, another way to get an error is through erroneous matches. And in the interest of time, I won't say anything about Josiah Sutton. I've written several pieces about this if you're interested in the case. I mean, he was falsely linked to a Houston rape, due basically to a misinterpretation of test results by the analyst. There was a way of disambiguating a mixture that the analyst failed to do. I mean, it's some indication of the level of work of the Houston Crime Lab that, when I looked at the case 5 years later, here I am, a professor in California, and the case was sent to me by a television station. And I looked through this and said, wait a minute, this guy, he might not be guilty. Oh my goodness. There's actually very good evidence that he's not a contributor to the mixture that the lab had him a contributor to. So it was just this kind of laboratory error can occur. A misinterpretation of mixtures can occur. And in the paper I mentioned a mixture misinterpretation case that led to a false cold hit in Sacramento. So we know that these kinds of things can cause false matches. I won't tell the Sutton story. So here we are with Sutton. When he was arrested, he'd just made the high school football team, and the team has this traditional. They all go out to a lobster dinner, and he never got it to his lobster dinner because he got arrested. And anyway, after we got him out of prison, I took him out for his lobster. So all right. So now, all right. So lab errors, and I mentioned earlier that the cross-contamination of samples and so on, and mix-ups. How often does that all happen? If you ask people who work in labs, they generally say very rare. I mean, human error, it can occur, but not in our lab because we're careful. We've never made an error, and if we ever did make an error, we would correct whatever caused us to make an error, and so then we wouldn't make an error again. So let me show you. This is a guideline issued by the FBI suggesting that labs keep corrective action files. In a number of labs, when asked if, I'll show you one particular lab that when asked by defense lawyers, do you keep corrective action files, said no need. We've never had an error. Right? Now, in this particular jurisdiction, which is Kern County, California, it's an accredited lab. They ran the file because later there was a case where, in order to incriminate the person they wanted to incriminate, they had to admit that they'd made an error at one point. And the defense lawyer said, well, wait a minute. Did you document this error as required by the FBI standards? Yes, we did. Well, where do you keep documentation of such errors? Oh, we keep it in our error file. Oh, so you have an error file. Can we see the error file? Well, earlier it had been called a corrective action file. Who knew? But turned out there was a file. Shall we take a look? These things come to me. I mean, so now mind you, this is the lab that's never had an error, and if they ever did, their error rate is 0, and if they ever did have an error, they would correct it so it would be 0 again, right? All right. So here we have cross-contamination of reference sample. This is the analyst's handwriting. Mother's sample contains minor profile, appears to be child's from same case, must reamp sample. All right. Positive results in a negative control. This is very, very common. Negative controls are samples that have no DNA in them supposedly, but peaks are showing up, right? Alleles are there. It's due to mix-up of tubes. Let's see. It's clear. Let's see. Tubes D2 and D4 were switched on accident. Accidents and the errors maybe want to happen. Here, analyst DNA contaminates sample. I like this. There's the known reference sample was contaminated with analyst Vicky Kump's DNA profile. It could not be determined at what stage of the analysis this contamination occurred. Peter Neufeld told a story, an interesting story about analyst contamination last night. It was interesting. Here is separate DNA extracts accidentally combined in the same tube. You want a false match? Here's a false match, right? So two separate samples are being extracted and accidentally get into the same tube. The analyst figured this out, that it happened, although you can imagine circumstances where it wouldn't be apparent that it happened, right? Anyway, the error leads to the so-called corrective action. I particularly like this corrective action. This is how they're going to make the error rate go back to 0. Try to minimize interruptions during any sample transfer procedure. Signs were made for the analyst to put at the end of their work area indicating they cannot be interrupted at the time the sign is out. Also, phone calls will not be answered during any sample handling and transfer steps. Okay, will that fix it? Is the error rate back to 0 again? Well, no. Samples mixed up again. All right, it goes on. I mean, this is just sort of a sampling. Oh, I like this one. Suspect tested twice, doesn't match himself. Poor Mr. Phillips, he wasn't himself that day. And look at the investigation down here. So they were asking. There was a sample labeling error here, and when they interrogated the people who were supposed to be labeling the samples, I like this. He responded that he did not expect to have to do someone else's job for them, and that they, the collection room staff, are always getting people's names wrong. So he only goes by the case number, not the name. In addition, he later told me, he fills out the sample identified. All right. So, all right. But this is the lab that has an error rate of 0. All right. Well, all right. I mean, so when you look very closely, it's like it's like they say about sausage. You don't want to look too closely. When you look closely, you start seeing these things that lead you to believe that there is a possibility of cross-contamination. Here's a case I ran across. I looked at some materials for a journalist from Seattle who got corrective action files from the Washington State Patrol. Here we have accidental cross-contamination causing a false cold hit to a juvenile sample. I'll just pull up the key thing. So the processing a rape case, unknown DNA shows up in the rape case, and they search this through the database. They find a hit to a felon. They took it out, but the felon was only a child when the crime occurred. It was determined, so how did this happen? It was determined the felon's sample was being used as training sample by another analyst. So wasn't even the same, a different analyst is working with the felon's, the child, the juvenile felon's sample, and somehow the DNA from that case got into the rape case, and hence a cold hit. So, I mean, these things can happen. I'm not saying they're frequent. We don't really know how frequent they are because labs are kind of closed about disclosing these things. One case, and often don't admit it. I mean, one case where I think we had good evidence that there was a cold hit due to cross-contamination is this case of Jayden Lesky, which I worked on in Victoria, Australia. The story, it was a high-profile case. A toddler disappeared under bizarre circumstances and was found dead, and the mother's boyfriend was tried for the case but was acquitted, largely because there was DNA from an unknown person found on what was thought to be a blood stain on the toddler's clothing. So there was this loose end in the trial of the mother's boyfriend who had custody of the child during the period when the child was thought to have disappeared. He was acquitted, but there was later a coronial investigation. A coroner's court was constituted to look into, the mother's boyfriend's acquitted, who did kill the child, and the fact that when the child disappeared, the house was vandalized and a severed pig's head with blood was smeared around. And there was a number of circumstances that made people think that this was worthy of investigation. Now, this unknown profile on the clothing was of an unknown female, and sometime the acquittal was in 1999, I think. But then in 2001 or 2002, this unknown profile on the toddler's clothing was searched through the Victoria State database, and it produced a cold hit. And it produced a cold hit to a mentally retarded rape victim, an institutionalized woman who had, by all accounts, never left the small town where she lived, which was hundreds of miles away from the crime scene, and nobody could find any possible connection. She was in the database because, her profile is in the database because she was an alleged crime victim. There was